All right, are there any questions from last time? So last time I was mentioning that units can bind to form a very long helix or a circular ring. And on the next slide, I have a picture of that. You have a bunch of protein subunits. Okay, the two of them, if they pair together, that forms a dimer, right? Then in another example, you have these units that are linked end to end, right? And they're forming a helix. In yet another example, starting from this kind of protein unit, right? You can get these units to bind to each other and form a ring. And there are many other possibilities too. In fact, structures such as viruses and ribosomes, right? And we'll talk more about ribosomes later because ribosomes are the protein making machines. These are actually built from a mixture of one or more types of proteins and RNA or DNA molecules. And if you take like a virus or a ribosome and you separate out the different components, you can purify the different components, and after that, if you just put them together, right, they will spontaneously assemble into the respective structure, the virus or the ribosome. Now, it is a fact that a helix is a common motif that is found in biological structures and results from similar units that are connected end to end, just like I showed you over here, right? That's a helix. Now, some other proteins have got elongated fibrous shapes, such as alpha keratin. This is the protein that is found in hair, right? Collagen is a protein that is found in skin, and then another protein is elastin, right? And there are lots more examples. We are not going to get into that. Right? Now, outside the cell, the proteins are often stabilized by covalent cross-linkages such as the disulfide bonds between cysteine side chains. And let me show you the picture, right? So if you have, so this is the cysteine side chain, sulfur and hydrogen, right? So you have the, polypeptide backbone here. There is a cysteine side chain. Similarly, you have the polypeptide backbone here, right? Maybe the same protein. It's a continuation of the same backbone, but you have these two cysteine side chains. Now, inside the cell, all right, that's an aqueous environment. The hydrogen concentration is high, okay? So it will stay like this. But when it goes out of the cell, the two hydrogens are eliminated and the two sulfur atoms are linked together by what is called a disulfide bond, right? So you see these disulfide bonding between cysteine side chains, and typically outside the cell where you don't have enough hydrogen, you get this kind of bonding, and this is like a staple. Like you fold a piece of paper, then you staple it, okay, to make it more permanent. So this disulfide bond, it doesn't change the original shape of the protein, but it gives stability to that shape. Okay. Yeah, go ahead. I didn't get you. The disulfide bond is the bond between two sulfur atoms. Okay. So this is a protein. Okay. This is a protein. These are two side chains. It happens to have two cysteine side chains. There may be other side chains too. Okay. No, this is just a protein. It's not even in, a protein is inside a cell. It cannot be in different cells. This one protein is inside that cell, or it may be crossing the cell membrane, right? You will not have one protein spanning many different cells, Yeah, it's probably inside one cell, okay? So we go back to the previous slide. So whatever I showed you in the picture over there, that's covered using text here. So extracellular proteins are often stabilized by covalent cross-linkages, such as the disulfide bonds between cysteine side chains. And these serve as atomic staples to stabilize the three-dimensional conformation of the protein outside the cell. Inside the cell, you don't need these staples, right? And not only that, because you have an excess of hydrogen inside the cell because there is water, okay? Water produces hydrogen ions, so that disulfide bond would be broken, right? So you will have the structure that I showed in the first picture on the left, right? Inside the cell, yeah. Intracellular means inside the cell, okay? Extracellular means outside the cell. Like extraterrestrial creatures, okay? That means they are outside this world, yeah. Yeah, extra means outside, intra means inside. All right, any other questions? Yeah, go ahead. Yeah, they serve as atomic staples to stabilize the three-dimensional conformation. Yes, yes, yes. The disulfide bond. Yes, yeah. Well, it would be outside the cell. It would not be inside the cell, because inside the cell, the hydrogen ion concentration is high. Okay. See, what will happen is, yeah. So inside the cell, the hydrogen ion concentration is high. So this will not remain like this. Hydrogen will get attached here, hydrogen will get attached here, and you'll have this conformation. Outside the cell, you don't have the watery environment, you don't have hydrogen ion concentration. So this is one among many possibilities, all right? You can have, I mean, it's just that the book discussed this one, so I'm highlighting this one, but there are probably other examples of that. We're not going through an exhaustive list. All right, any other questions? Okay, next we move on to what are called protein-ligand interactions. So proteins bind to other molecules, which are called their ligands, with great specificity. To ligate means to tie, okay? So a ligand means something that the protein sticks onto, right? So if you talk to a biologist, right, they're not going to say that some molecule that the protein sticks onto. They'll talk about proteins and ligands and substrates and so on, okay? So this is biological jargon. So this ligand, the molecule that the protein binds to, must fit precisely or exactly into a groove on the protein surface. This will allow for the protein and its ligand to be linked together strongly by large numbers of weak covalent bonds. They have to fit exactly into each other. If the fit is not perfect, then that's not the ligand for that protein. Okay, so they differentiate based on the shape. Okay, so this perfect kind of fitting enables the protein to pick out its ligand in a crowded environment, because there may be a lot of different molecules. Okay, but it's exactly looking for its ligand, right? So that will have exactly the right shape to bind into the groove in the protein. The region of a protein which associates with the ligand, again, this is terminology, it's called a binding site. Okay. Now, an example of proteins that have got really very strong binding properties are what are called antibodies or immunoglobulins. Okay, these are the molecules that actually give you immunity when you take shots. Okay, to get immunity, right? It's antibodies or immunoglobulins. They are a class of proteins that have a highly developed binding capacity. They are produced by the immune system in animals in response to foreign molecules, right? So each antibody binds to a particular target molecule tightly and either inactivates the target directly or marks it for destruction. It marks it out as a bad guy. Okay. Now antibodies are Y-shaped molecules, and I'm going to show you a picture in a minute, with two identical binding sites. And each site is complementary to a small portion of the surface of the target antigen molecule. So you look here, right? This is what a typical antibody will look like. It's a Y-shaped molecule. There are two heavy chains, okay? This is a peptide chain, right? Two heavy chains and two light chains here, okay? But the shape is like a Y. Now each antibody will have a very, very specific shape over here, and over here, okay? That shape will be geared exactly towards its target, right? So if there is some antigen, again, that's also probably going to be some protein, that will fit in exactly here. Only that antigen will fit in here. Nothing else is going to fit in. So it can pick up its antigen with great specificity, right? So when they give you shots, okay, they basically inject the antigen corresponding to this antibody. In reaction to that, your body produces lots of antibodies, okay, lots of these Ys. So that now later on, if the same antigen attacks you, you have a lot of foot soldiers on the ground, okay, that can go and catch that and basically get rid of that antigen. Okay, that's the principle behind immunotherapy, right? Immunization, or the flu shot, or whatever shots that you take, that's why it works. Okay, so this I covered already. Any questions? So next time you go to the doctor's office to take a flu shot or something, think about this Y-shaped molecule, right? Now, an individual animal can make billions of different antibody molecules, right? Because there are many different antigens. Like after a little kid is born, the first couple of months they take all kinds of shots, okay, because they don't have any antibodies. Okay, you're trying to give them the antigen to stimulate the production of antibodies. So each antibody molecule has got a distinct antigen binding site. Antibodies defend us against infection in at least two different ways. The first one is antibody and antigen aggregates. That means a combination of the Y-shaped molecule and the antigen that binds over there, right? They are ingested by phagocytic cells. So phagocytic cells are cells in the body that are like trash cleaners. They will go and eat up other cells. So if a cell is marked, if an antibody, and if an antigen is marked with an antibody, right, there'll be specialized cells that will go and eat that one up, okay, and clean it up, okay. So that's one mechanism. The other one is that if you have bacteria or viruses that get coated with antibodies, they are marked out as the bad guys, all right, and there are special proteins in the blood that will kill them, okay. So you mark out the bad guys using these antibodies. Now, if you look at animal cells, right, antibodies are made by a class of white blood cells, which are called the B cell lymphocytes or the B cells, right? And each B cell will carry a different membrane-bound antibody molecule on its surface, which serves as a receptor for recognizing a specific antigen, right? So so you have the cell, on the surface of the cell, you have this molecule. Let me just see if I can write on this slide or not. Okay, maybe I can show some picture here. Let me just see. No, I cannot. Okay. No, not on this one. Yeah, which one is that? Oh, this one, this bar. Okay, let me just try that. I've never tried to write on the slide. This one. Oh, the pen. This one, right? Okay. Okay, let me see. Yeah. So, okay, I have to go back a few slides. Oh no, if I select the pen, then it'll not let me go back. Okay, so let's just go back and go back to that particular page, right? Okay. So okay, so here let me select the pen and just try. Okay, so here, see what they're saying is that each B cell, right? So if you have a, well, okay, it works. So if you have a B cell, okay, it is going to have, so if you look at the membrane of that B cell, right, then you are going to have the antibody, that Y-shaped molecule will be on the membrane, okay, for a particular B cell. And that antibody molecule will have exactly the right shape for one specific antigen. So when the antigen comes and binds here, okay, remember I talked about transmembrane proteins, right? When something comes and binds over there, there is a signal transduction inside the cell. It will stimulate the cell to divide, okay? So when the cell divides, it produces more cells exactly at the same time, right? So it will produce another cell that will have the same antibody on it. Oops, sorry. What happened? I don't know, something happened. But anyway, it's going to produce more copies of itself, right? And so that is the principle behind immunotherapy, right? So when the appropriate antigen binds to the receptor, the B cell is stimulated to divide. So there's a signal transduction inside the cell. There is a cell division machinery that is stimulated. The cell makes many copies of itself, and it'll secrete large amounts of the same antibody, right, bound on the membranes of these different cells that are produced. And this is the phenomenon that is used to raise the level of antibodies in animals and forms the basis of immunization and immunotherapy. Right. Now, antibodies can also be used to purify molecules, because remember, an antibody is like a probe that is very, very specific to that antigen molecule, right? It has exactly the right shape only for a particular antigen molecule. So in fact, in immunoaffinity column chromatography, a mixture of A, let's say you have an antigen A and other molecules, you pass it through a column that contains beads coated with the anti-A antibodies, right? So what is going to happen is that A and the anti-A antibodies will attract each other, right? So those A molecules will get stuck, right, to those beads, right, while everything else will pass through the column, right? So the flow through the column can be discarded because you're interested only in molecules of A, and then the A molecules can be recovered by appropriately treating the beads, because the beads now have basically anti-A antibodies, right, in combination with A, right? So you can set the A free by using some reagents. Another example where antibodies are used to purify molecules is the technique that is known as immunoprecipitation, where a mixture of molecules containing A is treated with anti-A antibodies, right? So now A and anti-A antibodies will form one complex, okay, together. So now you can do centrifugation. Okay, you have this mixture, then you rotate it in a centrifuge at very high speed. All right, and then you can separate out the A and the A molecules along with the anti-A and antibodies, right, from the rest of the stuff by centrifugation, right, and then from there you can recover the A molecules, right. So this is the technique that is known as immunoprecipitation. So in a few minutes I will talk about, see here we have chromatography, right, then we talked about centrifugation. So in the next few minutes I will talk about each of these techniques, how that is carried out. And again, for me it's easy to talk because I'll just spend like 4 or 5 minutes, right, I'm giving you the big picture. But when people do that in the lab, you know, there is a specific protocol that they have to follow, and you know, it's a time-consuming thing, right? So that way in engineering, we are lucky. We spend some nights and all that working on projects, you know, but you don't have to go and feed your rats something and then give them a shot at 2 in the morning, okay, come back in the morning to collect more data. We don't have to do those kind of things, you know, but it is really labor intensive in biological research. Now, monoclonal antibodies are large quantities of a single type of antibody molecule, right? If you want a large amount of a particular antibody, right, how do you produce that? So in order to produce that, you can fuse a B cell, which is taken from an animal injected with antigen A. If you injected the animal with antigen A, then the B cell will have the antibody corresponding to A, right, on its surface, right? So you can fuse this cell with a cell of a B cell tumor, right? Like, you know, for example, lymphomas and all, those are B cell tumors, right? So the tumor will have the characteristic that the cell will keep on dividing, right? Because normally, as I said before, there is a dynamic equilibrium between the rate at which new cells are created in your body, right, versus the rate at which old cells are getting removed, right? But you get a tumor when this equilibrium is disrupted, okay? You have some cells that are just dividing even when no more new cells are needed, right? So you can take a cell from a tumor and that will have that characteristic that it will keep on dividing, but it doesn't make the antibody, right? But if you fuse it to the B cell, right, that is capable of making that antibody, right, then the combined cell, the resulting cell that you get, it has got both properties, okay? It can keep on dividing, right, and it can also make more of that antibody because every time that cell divides, more, you know, antibodies are being produced, right? So this resulting cell divides indefinitely and also produces the antibody, and it will be producing the same antibody over and over again. So you will call it a monoclonal antibody because you're going to have lots of that. Okay? It's monoclonal because it's only of one type. Okay. Now antibodies can also be used as molecular tags or molecular markers. For example, one could couple a set of antibody molecules to a fluorescent dye, right, to some color, right, or colloidal gold particle or some other special tag. So that way you'll be able to track, okay, if you have these antibodies that have been gotten this tag inserted on them, then you'll be able to track what is happening to those antibodies, okay. All right, so now in order to study proteins, right, now proteins are actually inside cells, so you have to first isolate them from cells and tissues, right. I mean, nobody is going to come and just give you some protein and study. You have to get it from the cell. Okay, so you have to isolate the protein that you're interested in from inside the cell. So now the cell has a cell membrane. So the first step for protein isolation is to break open the cell by disrupting its plasma membrane or cell membrane. And 4 different approaches are commonly used. The first one is to break cells with high frequency sound. Then use a mild detergent to make holes in the plasma membrane, right? That'll happen because, see, the cell membrane is made of lipid, right? I mean, if you have grease on your plate, okay, how do you get rid of that? You basically put some soap onto that, right? I mean, so if you use a detergent, you probably will make holes in the plasma membrane, right? Another one is to force cells through a small hole using high pressure. So that's a mechanical procedure. You'll rupture the cell membrane, right? Yet another method is to shear cells between a close-fitting rotating plunger and the thick walls of a glass vessel. And again, I mean, I'm you know, mentioning each of these techniques as just like one sentence, right, or a phrase. To actually do it will require some expertise, right? You have to have hands-on experience in running these biological procedures, okay? To make sure that you are getting what you want and you don't mess up everything. You don't mess up the biological sample. But once you have done that, right, then what you're left with, so you've broken the cell membrane, the stuff inside comes out, which is called the homogenate or abstract. It contains molecules from the cytosol, right? And recall, the cytosol is basically the inside of the cell with all the organelles taken out, right? So So the homogenate contains the cytosol as well as the membrane-bounded organelles, like maybe the nucleus, the mitochondrion, the chloroplast, and so on, peroxisome and lysosome, and so on. Right? Now, if you're careful when you're breaking the cell membrane, right, you probably would not have messed up. And there's a protocol. I mean, there will be instructions from the manufacturer of different equipment that you use, what steps to follow, for how long to do it, at what speed, and all, to make sure that you don't break up the stuff inside. So if you're careful, then that can leave the membrane-bounded organelles intact, so you don't mess up like the nucleus and other things, right? So the next step is to separate out all these different components, right? Because you're interested in studying, let's say, some proteins. You're not interested in the nucleus. You're not interested in the let's say, Golgi apparatus or whatever. Okay, so how do you separate out the components? There are a number of procedures that can be used for separation, such as centrifugation, then velocity sedimentation, and equilibrium sedimentation. And I will discuss these one by one, right? And you can also use column chromatography, as I mentioned earlier, and I'm going to give you details on that in the next few slides, right? So first, so we are going to look at centrifugation, velocity sedimentation, equilibrium sedimentation, right? And then we'll also look at column chromatography, right? So first, let's look at centrifugation, right? So what is done in centrifugation? You basically take the abstract that you have, put it in a test tube, and rotate it at very high speeds. And very high means really high. It's like 10,000. The acceleration will be like 10,000 times the acceleration due to gravity. So you can guess that if you're centrifuging something, okay, it better be inside a very tight chamber, right? A lot of heat is going to be generated, right? So it has to be well protected, right? That's why, you know, for people that work in these labs, okay, they have to follow a lot of precautions, because if that thing comes out and hits you, you're going to be dead. Okay? It's like going to be like a bullet, right? So the homogenate is placed in test tubes and it's rotated at very high speeds in a centrifuge, right? And the components will separate out mainly on the basis of their size and density, right? Remember mv square over r or whatever, the forces that are acting, right? They will separate them out on the basis of their size and density. Now, you could repeat the procedure. You could initially rotate it at a particular speed, get some separation, right? Collect the stuff at the bottom. Then whatever is left behind, liquid is left behind, put it in another test tube, again rotate it at a different speed, get further separation. So you can repeat the procedure, right? So repeated centrifugation at progressively higher speeds will fractionate the cell homogenate into its different components. And of course, as a biologist, I'm saying, yeah, do it at some speed and then next higher speed, but you have to know what speed exactly it is going to be, okay, to get the separation that you want. So if you have a test tube at the bottom, you'll get the pellet, right? And the separated thing, you can take it out, then take this liquid that is still there and do it again to get further separation. Now, another technique is what is called velocity sedimentation. And this is based on the fact that the subcellular components, that means the components inside a cell, like the nucleus, the mitochondrion, the chloroplast, and so on, these will go at different speeds, right? So subcellular components are carefully layered over a dilute salt solution, okay? And as they go through the solution under the effect of gravity, they will go at different speeds, okay? So as a result, some bands will be formed. So these components sediment at different speeds according to their size. When sedimented through such a dilute solution, different cell components will separate out into distinct bands, right, in the test tube. So you can make a hole in the test tube at different locations and then collect the different components that you're interested in. So after an appropriate time, the components can be collected individually. Another procedure is called equilibrium sedimentation. This procedure can be used to separate cellular components on the basis of what is called their Bowen density, independently of the shape or size. And this makes use of a centrifuge. The sample is usually going to be layered on top of a steep density gradient that contains a very high concentration of sucrose or cesium chloride. Okay, I don't know why they use these, because that probably is good for setting up that gradient, but that's the standard that is used. So now you've done that. Okay? You've put the sample on top of this high concentration sucrose or cesium chloride. So you have a density gradient. Now you centrifuge this test tube, right? So things will keep on moving until they reach a place where the density of that molecule is the same as the density of the surrounding medium. Then it doesn't move anymore. So each subcellular component will move up or down when centrifuged until it reaches a position where its density matches that of its surrounding, and then it'll move no more. So a series of distinct bands will form inside the test tubes, and these bands can be separated out by collecting from the base of the test tube. So you have separated out the components. And again, I mean, we don't need to know the details because we are not trying to learn biology, but have some idea, you know, when they refer to all these different procedures, right, in the materials and methods sections of the papers, right, you should have an idea about what they're talking about, right? You would not know the details, okay, for how long do you have to do it, okay? How many bands are going to be produced, you know? I don't need to know that, you don't need to know that either, but at least it's good to have some idea. All right, any questions so far? So we now continue with separating a mixture of proteins. See, proteins are very diverse molecules. They differ from each other by size, shape, charge, hydrophobicity, etc. And you can use all of these properties to separate proteins from each other so that they can be studied individually. Right? And the two techniques that are commonly used, one of them is called column chromatography, and the other one is called gel electrophoresis. Right? These are experimental procedures that can be used to separate out a mixture of proteins. Right? And let's look at them one by one. So in column chromatography, the mixture of proteins is forced through a vertical column that contains some kind of a matrix. Right? The sample is positioned on top of the matrix, and solvent from a reservoir is used to force the sample down the matrix. And different choices for the matrix will lead to different kinds of chromatography. Right? So I think I have a picture here. Right? So let's look at this picture. So you have a column here, there's a solid matrix. You'll put the sample on this, okay? And then they put some liquid solvent on it, right? To force the sample to go through this solid matrix, right? And depending on the type of column that the matrix that you have, you'll have different kinds of chromatography, right? So let's go to the previous slide. So if you have ion exchange chromatography, then the matrix that you'll use in that column will consist of positively or negatively charged beads, which will have the property that it will retard proteins of the opposite charge, right? I mean, you know that positive charges attract negative charges and vice versa, right? So if you have positively charged beads in that column, then all the negatively charged proteins will get attached, right, to the positively charged beads, right? And everything else will go through, right? And then later on, you can release these positively charged proteins, right? And you achieve separation. So separation in this case is achieved on the basis of charge, right? Then in another case, when you have gel filtration chromatography, the matrix consists of small porous beads, right? So again, here the separation will be achieved on the basis of size. Protein molecules that are small enough to get inside those beads, they'll get stuck in the column, right? They're not going to go through, right? So protein molecules that are small enough to enter the holes in the beads are delayed and travel more slowly through the column. And in this case, again, the separation is achieved on the basis of size. So for the first one, it was on the basis of charge. The next one is on the basis of size. Yet another procedure that you can use for separating proteins is by using like the antibody kind of property, right? If the matrix has got antibodies for a particular molecule, when you push the sample through that column, right, the antibodies will pick up those molecules because they bind very tightly to them. Everything else will go through. Okay, so that is referred to as affinity chromatography. So in affinity chromatography, the matrix will consist of molecules which interact specifically with the protein of interest, for example, an antibody. So these are the three main types of chromatography that are extensively used. Now in all three of them, basically you're trying to get the proteins that you're interested in to bind to the matrix, whether it is because of opposite charge, whether it's because of size, whether it's because of antibody properties, right? Antibody antigen binding. But then later on you need to separate out the proteins that have gotten stuck to this solid matrix. So that can be done by a pH change or by concentrated salt solutions. And the proteins which bind specifically to such a column can be finally released by this pH change or by concentrated salt solutions, and then they emerge highly purified. All right, any questions? So one procedure is column chromatography, right? Yet another procedure is what is called gel electrophoresis, right? And this should be close to the hearts of EEs because here electric field is used, okay? The idea is, you know, if you take a protein, you mix it up with something, make it a negatively charged particle, right? Then you subject it to an electric field. That is going to move, right? And the amount by which it will move will depend on the size of the protein, right? So different proteins will move through different amounts, right? That's the whole idea in gel electrophoresis. So this procedure is used to separate proteins on the basis of their size and net charge. So if you're separating on the basis of size, then you use what is called SDS page, right? So SDS is abbreviation for sodium dodecyl sulfate polyacrylamide gel, right? So, I'm sorry, SDS is sodium dodecyl sulfate, right? And page is polyacrylamide gel electrophoresis, right? See, those of you that took the lab, I mean the tour of the lab yesterday, some of you went, right? I mean, to the center and got a tour of the lab. I think they showed you something about electrophoresis, you know, because when you went to that other room, they showed you there was something where you could actually move the electrode, right, the positive charge, and get it to move from one point to the other. And that is gel electrophoresis that they showed you. Like many of you went, right, yesterday or no? I think many of you went. Those of you that went, you did get to see it. Right? So in the case of sodium dodecyl sulfate polyacrylamide gel electrophoresis, protein separation is achieved on the basis of size, because you've mixed it up with SDS, right, which is negatively charged, right? So under an electric field it is going to move, but the amount by which it will move will depend on the size of that particular protein. If the protein is smaller in size, it has a smaller molecular weight, it will migrate more. If it is larger in size, it will migrate less. So you can separate out proteins on the basis of size. Now in the case of, there is another technique where you can separate proteins out on the basis of charge, and that technique is called isoelectric focusing. So I'm going to discuss that after I give you some more details about SDS page, then I will discuss this one also. And then we'll see how you can actually separate proteins in two perpendicular directions. In one direction on the basis of size, right? And then in a perpendicular direction on the basis of electric charge, right? To achieve further separation. So let's first talk about SDS page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis. So in this, the individual polypeptide chains will form a complex with negatively charged molecules of SDS. So the whole thing becomes negatively charged. When subjected to an electric field, they will migrate as a negatively charged SDS protein complex through a slab of porous polyacrylamide gel. Now the proteins will migrate at a rate which is proportional to their molecular weight. Thus protein separation is on the basis of their size. Okay, so this is SDS page. Now on the other hand, if you want to do isoelectric focusing, you're going to separate proteins on the basis of their charge. Now to do that, we note that for any protein, there is a characteristic pH value, right? pH is the hydrogen ion concentration. So it's basically, you know, a measure of the charge, right, in the medium. So there is a characteristic pH value called the isoelectric point, at which the protein has no net charge and therefore will not move in an electric field, right? So if you have an electric field, right, if the protein gets to the region where, you know, the charge on it is the same as the charge on the surroundings, it's not going to move anymore, right? So in isoelectric focusing, proteins are electrophoresed in a narrow tube of polyacrylamide gel in which you have established a pH gradient by a mixture of special buffers. So each protein will move to a point in the gradient that will correspond to its isoelectric point, right, where the surrounding pH is basically matching up with the charge on it, and it will stay there, thus allowing protein separation on the basis of charge. And now if you want to separate out proteins both on the basis of size and charge, you can run this in two perpendicular directions. So this method, the two-dimensional polyacrylamide gel electrophoresis, combines the two procedures just discussed to separate proteins on the basis of both size and electric charge. In this method, first you will run isoelectric focusing, is used to separate proteins on the basis of electric charge, right? So in a particular direction. Then you apply this SDS, or sodium dodecyl sulfate page polyacrylamide gel electrophoresis, in a perpendicular direction, right? So that in that direction you're now separating it out on the basis of size, right? So you achieve more separation of the proteins. So this is the picture for that. So in one direction you have applied the stable pH gradient, right? The other direction you're running SDS page. So this side there is separation that is based on charge, like this and this guy have been separated based on charge, but then the separation of this one and this one in this direction is due to the size of the proteins, right? So this is two-dimensional polyacrylamide gel electrophoresis. And typically, all right, once you separate this stuff out, okay, you probably might use some blotting paper, right, to capture these spots, okay. So that's why you'll hear biologists talk about western blot, southern blot, northern blot, and so on. Basically, they would put blotting paper on here, capture that information. And now they will subject it to further analysis. Okay, so that they can use this equipment, this SDS page or this isoelectric focusing equipment to run additional experiments. So the information will be captured on some blotting paper, right? And the equipment they showed you in the lab yesterday, that doesn't need that blotting paper, because by moving things around, you're making sure you're moving the electrode, right? So you're already collecting it. You're basically collecting this information from here by putting some electrodes at those locations. Okay, that's what they showed. Yeah, go ahead.
Is there any relation between the gradient and the SDS stage?
Not really, no. Yeah. You mean this pH? No, the gradient is basically you're separating it on the basis of charge, electric charge. SDS page is basically you link it up with a negatively charged molecule, and you're saying that the amount by which it will go in this direction, right, will depend on the size of the protein that you have. So if it's a smaller protein, it'll be probably higher up. The larger one will be going further down. Okay. But you could have a situation where two proteins have the same size, so they would be at the same location. But then you go in this direction, right, and then separate them out on the basis of charge. Okay. All right, any questions? Now today I sent you guys quite a few links and all that. It's not required material for this class, but people who are doing research in this area, like the AgriLife students and all, you might want to start looking at those, right? There's tons of information over there, right? Maybe you don't have time to read everything, but it'll be useful. And I sent it to the whole class because who knows, there might be other people in this class who get interested in this area. Especially if they're also taking Pengyu's course, right? So. Now, how do you determine protein structure? So remember, protein structure, we said that there were different levels of protein structure. The primary structure was the amino acid sequence. The secondary structure was the presence of alpha helices and beta sheets. The tertiary structure was basically the three-dimensional structure. And then we talked about the quaternary structure, which is for proteins that are made up of more than one polypeptide chain. Okay? So the question is, how do you determine the structure of a protein? There are a number of different approaches. So, and here we are talking about initially at least the primary structure. So there are a number of different approaches that can be used to determine protein structure. In the first approach, you add a reagent that can cut off. See, like if you have a protein, right, it's basically a bunch of amino acids that are linked together. And then you have an amino terminus or an N terminus on one end and a carboxyl terminus on the other, right? So the first approach, the most obvious approach is if you can get a reagent that'll go and cut off the amino acid from the amino terminus, okay, the first amino acid. Then you can go and figure out, where it goes under gel electrophoresis, and you can figure out what that particular amino acid is. So in the first approach, a reagent is used to isolate the N-terminus amino acid from the rest of the polypeptide chain. The isolated amino acid can then be identified using a full set of known amino acid derivative standards. Example, the position to which a particular amino acid migrates onto SDS page. You have 20 different amino acids. So maybe they go to 20 different positions. Okay, so you match it up and you say, okay, the first amino acid that I cut out, it migrated to this position. Maybe it is lysine, it is arginine, or whatever, wherever it matches up. And you can keep on repeating this process. Like if you have a protein that is 20 amino acids long, first cut out the first amino acids. See where it migrates. Figure out, okay, what amino acid that is. Whether it's histidine, tyrosine, whatever. Okay, then cut out the second one, and so on. Keep doing it. Do it 20 times. It's a laborious procedure. Okay. So this process can be repeatedly used to determine the entire amino acid sequence of the protein. However, many times it is found that the amino terminus amino acid is chemically blocked, right? You cannot use the reagent to cut that out, right? Because, the cell has produced the protein for a particular purpose, okay? It hasn't produced the protein so that I can go in there with a reagent and cut it out, okay? So it'll have its own defensive mechanism. So it might be blocked, okay? Maybe the chemical that I have will not go and cut it out. So in that case, you cannot use this particular method, right? Because the amino terminus amino acid is chemically blocked. So it cannot react with anything. Now the second approach to find out, protein structure or the primary sequence of amino acids is it makes use of the fact that there are certain chemicals which are known to selectively cut proteins at particular amino acid locations. For example, there is an enzyme that is called trypsin, which will cut on the carboxyl side of lysine, right? So if you have a lysine in that protein chain, you have lysine, okay? There, on one side it was the amino side, the other side is the carboxyl side. So on the carboxyl side of lysine, if you treat this protein with that enzyme, wherever there is a lysine, on the carboxyl side of that is going to go and cut it, right? Similarly, there is another reagent that is called cyanogen bromide, which will cut peptide bonds on the carboxyl side of methionine. This is another amino acid, right? And probably there are more enzymes available. So what you could do is, instead of trying to, get the amino acid sequence for the entire protein in one shot, you could treat it with, you could take one sample, treat it with a reagent that is trypsin, right? That'll produce smaller peptides, okay? And then run them through gel electrophoresis. Do the same thing with other reagents, for example, cyanogen bromide, right? Do it again, you'll get another map, okay? And then try to, put together all this information, right, to figure out what the original protein is. So these reagents can produce a few relatively large peptides. These can be separated using, for instance, gel electrophoresis to create a peptide map, which is diagnostic of the protein from which the peptides are generated. And then the sequencing of these overlapping peptides can be used to laboriously piece together the sequence of the original protein. Again, it's not a clear-cut procedure, right? But, it's like if you have a long thing, you don't know what it is, you break it up into pieces and try to find out some of the individual pieces, then, there is overlapping stuff, so try to put that together, And the statisticians and the mathematicians will worry about how many times you have to do it and all that to get this thing correct, okay? Now the third approach is something that I won't be able to explain fully as yet because we have not covered DNA technology, right? But in this approach, see, the DNA codes for proteins, okay? The DNA in the living organism codes for proteins, okay? And there's something called the genetic code, because remember in DNA you have 4 letters, A, G, C, T, okay? Now it can be shown that any 3 combinations of letters, they code for an amino acid, right? It can be shown. So what you could do is, if you knew the sequence of as few as 20 amino acids in a protein, right? So let's say there's a large protein that has got 200 amino acids, but let's say 20 of them, okay, or a few of them. So what you could do is you could go and construct a DNA probe, right? Based on the amino acids that are present, okay, you know what the corresponding DNA code will be. It'll be a small DNA code, but with that you can go fishing for the much larger segment of DNA that codes for that protein. Maybe thousands of base pairs, okay, thousands of these nucleotides are coding for that protein. But if you are able to figure out a few of them, right, you can come up with a probe, and DNA has got the property, right, that A always links up with T, G links up with C, okay, so you can go with a probe. It's like doing ones and twos complement, okay, twos complement, like wherever there's 1, there's 0 and so on, okay, so if you use the complement, you can go fishing for the original thing, right, and you don't need the entire complement. If I know some amount, okay, I can go and fish. If there's a match here, then I know, hey, this is the partner. This is the entire, sequence that contributed to this protein. And now I can look at that sequence and I can translate it, 3 at a time and find out what those amino acids are. So this part, if you don't understand fully, that's fine. I can talk about this later on once we have covered DNA technology. So we note that knowing the sequence of as few as 20 amino acids of a protein is often enough to allow a DNA probe to be designed so that the gene encoding the protein can be cloned. Right? Clone means many copies can be made. Once the gene or the corresponding complementary DNA, again this is Greek to you right now, has been sequenced, the rest of the protein's amino acid sequence can be deduced by reference to the genetic code. The genetic code is going to tell you which 3 letters map into which amino acid. Right? It's a degenerate code, but it's a universal code. It's used in all organisms. And the people that found the genetic code, they got Nobel Prizes in the 60s. Yeah. Now, in addition to these are about determining the primary structure of a protein, the amino acid sequence. In addition to these above approaches, the 3-dimensional structure of protein molecules has been studied using X-ray crystallography and nuclear magnetic resonance spectroscopy, right? And there are physicists that do that in the physics department, right? They want to know what the 3-dimensional shape of the protein will be. As I told you before, it's not an easy problem to go from the amino acid sequence to the 3-dimensional thing. But once you have the protein that you want and you want to study its properties, its structure, you can use other techniques like the physics guys do, right? They're not trying to make a prediction, they're trying to see what the structure is. Or if you can change some amino acids to change the structure a little bit, give it slightly different property, right? You might have a drug that was not working that well, right? You fine-tuned it and you really got something that works really, really well, okay? Because most of the time, many of these drugs, these are proteins. Okay, they are supposed to go and bind something, and either activate some process or inactivate some process. So for the binding to take place, they have to have exactly the right shape, right? And so shape is everything. All right, are there any questions? Okay, now we'll talk about proteins that are enzymes, right? And I had actually intended to cover only up to here today. So do you want me to cover a little bit more, or you don't care? I mean, maybe I went faster than I thought I would go, you know, so. But I can talk about this, you know, so. Or you have any questions or no? Yeah, let's talk about this, okay? I mean, it wouldn't hurt to, you know, get a little bit ahead. So proteins that are enzymes, okay? We said before, proteins can function as catalysts, okay? And as very, very powerful catalysts. There are enzymes that can speed up reactions by a factor of 10 to the, even as much as 10 to the power of 14. Now the binding strength between an enzyme and its substrate, right? Again, this is terminology. Enzyme is a protein which is a catalyst, right? The molecule that it acts on, it could also be a protein. That is called its substrate, right? So the binding strength between an enzyme and its substrate, or you know, an antibody will interact with an antigen. It is measured by the equilibrium constant K, right? So consider the reaction. Let's say two things react together. A and B react together to give you AB, right? So this is an association reaction. So for this reaction, the reaction rate, which is called the association rate, is given by association rate is some constant, association rate constant, times the product of the concentrations of the reactants. Okay, so this is the law of I think it's called the law of mass action, right? In chemistry or chemical engineering, right? The rate at which the reaction happens is proportional to the concentration of the reactants. But remember, the reaction can go in both directions. The reaction going this way is proportional to the concentration of these two guys. So it will be some constant times the concentration of A times the concentration of B. The reaction going in the reverse direction, AB becoming A plus B, that will be proportional to some other constant times the concentration of the reactant, in this case, which is AB, right? Because that's the reaction going in that direction. And at equilibrium, the association rate will become equal to the dissociation rate, right? And that's an equilibrium when the concentration of AB, everything stays the same, does not change. So the reverse reaction is called dissociation, where AB is dissociating to give you A plus B. And for this reaction, the dissociation rate is given by dissociation rate equals the dissociation rate constant K off times the concentration of the reactant in this case, which is AB, which is the product AB. At equilibrium, these two things should be equal, right? The association rate, so as much of AB should be produced, right, as is being broken up to produce A plus B, right? So the association rate should be equal to the dissociation rate. So you'll have K on times the equilibrium concentration of A, equilibrium concentration of B will be equal to K off times the equilibrium concentration of AB, right? Now this quantity, right, the equilibrium concentration of AB divided by the equilibrium concentration of A times the equilibrium concentration of E, that is called the equilibrium constant, right? This quantity K, AB equilibrium divided by the equilibrium concentration of A times the equilibrium concentration of B is called the equilibrium constant. Now if K is large, right, if the equilibrium constant is large, then you can see if this is large, then the AB equilibrium, the concentration of the product is much higher than the concentration of the individual reactants, equilibrium concentration, which means that the reaction will move very strongly in the forward direction, right? So the binding between A and B is very strong. The free energy of AB minus the free energy of A plus B is very negative, and the reaction is dominant in the forward direction. Right? Makes sense, and is in accordance with the second law of thermodynamics. Now enzymes, as we said, they are powerful and highly specific catalysts, and each enzyme will catalyze only one very specific chemical reactions. And enzymes that catalyze similar reactions, they can be grouped together and they have special names, you know. They have all kinds of names, and I'll get to that next time. You know, hydrolases are enzymes which will basically carry out a hydrolytic cleavage reaction. That means you add a water molecule and you break something up to carry out hydrolysis. Nucleases are enzymes that break down nucleic acids. Okay, so let me just stop on this side because there is an exhaustive list. I think even on the next slide of many of these different enzymes, so we'll talk about these, the listing of the enzymes next time. Okay, different kinds of enzymes that have properties. And enzymes, basically they're responsible for all cellular functions. If you do not have the enzyme that is needed to digest milk, you are in trouble, okay? That enzyme is called lactase. Your body needs to produce lactase, right? If your body doesn't produce lactase, that's when you have lactose intolerance. You drink a cup of milk and, you might even end up in the hospital, okay? So enzymes are very much needed. All right, any questions? Then let's meet again on Monday, okay?